Awesome. Welcome to all of our 2020 walkers. I have the honor today of chatting with some real superstars in our community. This is a crew of superheroes that needs no introduction. However, for those of you who are newer to the community or maybe haven't put faces to names just yet, I am joined today by Dr. Stormy Chamberlain, Dr. Mark Zilka, and Dr. Ben Philpott. Who really have contributed much of what we know about Angelman syndrome, how Angelman syndrome affects the body, and including really pivotal work, proof of concept work telling us that reactivating the paternal allele or dad's silent copy of UBE3A can be a possible therapeutic treatment, i.e. the therapeutic treatments that are coming into clinical trials in 2020. So the brains behind this virtual group is really astounding. And so what we're gonna do today is not what we typically do. So if you guys are interested in learning more about what this group of scientists has done, what your donor dollars have funded, you can go to the Angelman Syndrome Foundation website to follow links to their web pages, but also to see historically what the foundation has funded and what projects have come out of that funding. But what we're going to do today is a little bit different. We are going to get to know the people behind the science a little bit. So I'm going to lob a few questions at these guys. We're so thankful for their time today, but we want to get to know you guys a little bit better and know what got you interested in researching Angelman syndrome. And for this first question, what are some things that you like to do when you're not in the lab? So I'm going to throw this out to Dr. Ben Philpott first. Who is Ben Philpott? What got you interested in researching Angelman syndrome, and what are some things that you like to do for fun?
Yeah, so let's see, what got me into Angelman syndrome? Well, it actually started from a collaboration with Mike Ehlers, who was then at Duke University, and he convinced me to work on a collaboration with him on Angelman syndrome. And I got really excited about the research. But it really was when I met the parents that I think was in, somewhere in a 2006 to 2008 meeting that was up in Boston. And I got to meet some parents like Susan Ravelette, Lisa Franco, Eileen Brown, and it just really drew me into the community. And I was just so thrilled to start meeting individuals with Angelman syndrome and to learn more about the community. And that's what really got me excited about it. And then the other question was, what do I do when I'm not studying Angelman syndrome research? I like to travel. So that's a huge joy of mine, is to see new communities and new cultures and just to travel the world and experience life in different ways.
Very cool. So is it fair to say that you've been doing Angelman syndrome research for almost 15 years?
Yeah, it is fair to say that. Yeah, exactly.
Okay. Do you remember when the first grant that you received for Angelman syndrome was?
I probably should know this, but I don't. I'm guessing it was somewhere around 2008 or somewhere around there.
Okay, great. So we have like a decade and a half of your life dedicated to Angelman syndrome for Ben Philpott. Okay, who is Stormy Chamberlain? What got you interested in researching Angelman syndrome, and what are some things that you like to do?
So I came at this a little bit differently than Ben. As an undergraduate student, I learned about genomic imprinting from an awesome bad A female scientist who studied imprinting, Shirley Tillman. And I got excited about the prospect that there are some genes that are made only from mom's copy of chromosomes, some genes made only from dad's copy. So I did my graduate work at the University of Florida, where we studied Prader-Willi syndrome, which many of you might know as somewhat of a sister disorder to Angelman syndrome. And it was there that I got to focus a little bit on Angelman syndrome, trying to understand how the antisense transcript was regulated. So I can't tell you when my first grant was published, when I got my first grant on Angelman syndrome either. I'm going to guess it's like 2007 or 2008. But I can tell you when my first paper was published, and that was like 2001.
Oh my goodness, wow.
So a long time ago, before I sort of knew what I was doing.
So almost 2 decades here.
Yeah, maybe.
We're at a cumulative 35 years of your research careers. Go on.
So what do I like to do for fun? Well, I've learned recently that I'm probably an 8-year-old boy trapped in the body of a 40-something-year-old woman. So I enjoy running. I enjoy fishing. I enjoy hanging out with my kids, watching their sports. That's what I do outside of work.
Okay. All right. Very cool. So, all right. So we're keeping track here. We're at almost 35 years of research, if not more. All right. Who is Mark Zilka? What got you interested in this field? What keeps you here? And what are some fun things that you like to do?
Yeah. Well, I was just thinking about how 14 years ago is when I started at UNC. As an assistant professor, it's pretty different from college or any other school. Usually you have a whole class of people that you go to school with, but typically when you start as an assistant professor, there's nobody. It's just you, and you're sort of like navigating the world all by yourself. But I was very fortunate in that Ben had actually started at UNC about a year and a half before me. So he essentially had a very small class of me and Ben as assistant professors trying to navigate the scientific world. And so we would have lunch pretty much every day, talking about our science. I was working in the area of pain, still do. And Ben was obviously very interested in plasticity of the brain, learning and memory. So we would talk science. And as Ben had mentioned, he eventually got interested in studying Angelman syndrome. And then our conversation started turning to the Angelman syndrome and what it is. And it was just kind of fascinating to me to hear about the disorder and possible ways of treating it. And he was constantly hearing from the parents about the sensory abnormalities in the kids, like altered perception of warmth and temperature, and was trying to get me to study some of these features in the Angelman model mice, because that's something our lab really did well. I eventually relented, and I was like, all right, fine, we'll do this. We'll see if there are any sensory abnormalities in the Angelman mice. Wrote a grant to ASF, which was rejected. Did not get the grant to look at this. Nonetheless, we did find some issues with the mice. That paper ultimately got published much later because in the process, Ben, with these discussions, Ben and I were asking these questions as to whether or not you could reactivate the paternal allele, and if so, how would you do it? And so that initial discussion over lunch led to this interest in trying to figure out how to turn the paternal allele on. And that led to the work that we did with Brian Roth, identifying topoisomerase inhibitors. And ever since, I've now gone to the meetings, and I just realized that this is an amazing community. And I really felt that the work that we were doing in the lab was making an impact. I study pain at the other part of my lab. There obviously a lot of people suffer from chronic pain, but very rarely do we ever get to meet people who are suffering from chronic pain on a daily basis. But being able to go to these ASF meetings, meet the parents, the children, the scientists, and just really see how, without the work that we were doing, really nothing would get done in this area. So we really felt like the effort we were putting into this was making a difference.
Wow. Well, so I've heard about these epic lunches. I don't know if they seemed epic at the time, but I've heard about this for years, and I would have loved to have been a fly on the wall or under the umbrella I can imagine you guys outside that old research building on UNC's campus. But so fair to say you've been in this game for also another 10, 15 years, 12 years?
Sure, you could probably add maybe 10 to your tally.
Right. So we're close to 45, 50 years of combined research brain power here, which is really, really, really amazing. And you're absolutely right. I mean, this has contributed. This has gotten us to where we are today in 2020 with these clinical trials coming down the pipeline with the goal of unsilencing that paternal allele. So an idea that originated around a picnic table between you guys is really phenomenal. So I know, we tell you that all day, every day, because this is what impacts our life so dramatically. But you guys really are our superheroes. You are the superheroes to our community. So Ben can vouch for this. My question to him always has been, and probably continue to always will be, when I sit down for coffee with him, is what excites you now? What are you excited about right now? What should I be excited about as a parent? What research is happening right now that I should be excited about? So I'm going to pose this question back out to you guys. So Ben, what's exciting to you right now?
I think we're just seeing the fruition of a lot of hard work in terms of clinical trials, and this is extremely exciting. So, we have 3 groups that are putting out trials for the antisense oligonucleotides to reactivate the sign allele, paternal allele of UB3A, and that's incredibly exciting. This has just great translational potential. There are also small molecule therapeutics, such as the one that Ovid has. And other small molecule therapeutics that are being developed, including ones from our lab that might be able to reactivate the paternal allele of UB3A, and also a slew of gene therapies that are in development. And so it just makes it an incredibly exciting time, because there's been years and years of hard work. A lot of it started by people like Art Bodette and Joe Wagstaff, and now all this collective hard work from many scientists is now coming to the point where there are clinical trials, and that's just really exciting to me.
Awesome. Mark, what excites you?
I mean, basically the same thing. I've sort of been able to be part of this adventure, if you will. When I got involved in Angelman syndrome, we obviously knew about the gene. People were using treatments that would be beneficial for some of the symptoms, like seizures, but there really were no what we call disease-modifying treatments, even on the horizon. It wasn't even clear if that would be possible. And so in a relatively short period of time, we've gone from just identifying the gene, understanding what it does, to having these treatments that Ben was talking about. And I think that's pretty amazing. It really is a testament to how hard work, scientific research can really pay massive dividends to the point where now going to be, people are now being treated with the disease-modifying therapies.
Can't agree more. As a parent of a kid with Angelman syndrome, I can't agree more. I mean, again, I always walk away from my meetings with you guys feeling, re-energized and focused that we have so much to be hopeful for, but it's really nice to hear it from your mouth. So
Yeah.
Stormy, what are you most excited about?
Well, so in an effort to not say ditto, I'll say that, reflecting back on, how research that we invested in a long time ago has now materialized into therapeutics. I think about some of the research that we're investing in now, and where are we going to be in 5 to 10 years? Because if we think this is great, I think it's going to get better based on some of the things that we've invested in. Things like understanding the structure of UBE3A. I think there's going to be some novel therapeutics and some things that might help make the ASOs and other activation strategies or gene therapies work even better. On top of being able to help everybody, to be able to improve the lives of all individuals with Angelman syndrome, regardless of their age, their genetic etiology, and things like that. And so I kind of think that, we can now see how investments in research can pay off, and we've continued to invest in new forms of research with the idea that in the future it's going to look even better. And I'm excited about that, because we've realized that we've proven that can happen. So what's next?
I know in a lot of ways it feels like we're just at the starting line of an incredible race right now, right? In a lot of ways it feels like we've made it because we have these therapeutics that are coming down, and are actually have come to fruition in 2020, which is incredible. But you're exactly right. The things that we're investing in now, what kind of new and additional and adjunctive therapies are we going to be looking at 10 to 12, 15 years down the line based off of research investments now?
Absolutely. Absolutely.
Awesome. Anybody want to tackle the bonus question of what the ASF has meant to you?
I mean, the ASF gave me my start, right? I mean, it was through these meetings that I got to know the community. It was through these meetings I learned about funding opportunities and got pilot grant from the Angelman Syndrome Foundation. If it weren't for those initial pilot grants, I wouldn't be doing Angelman Syndrome research today, but that allowed me to get funding from other organizations such as the Science Foundation, and of course from NIH, allowed me to expand the research program. So it's really been a central focus of my career. It's been a great joy to be in the community.
And I would want to second that as well. I mean, I think, maybe parents don't, really recognize this. Obviously, the NIH and other funding agencies have, lots of money to give out for grants, but it's very difficult for a scientist to actually get those grants without, in many cases, an overwhelming amount of preliminary data. And so you're kind of stuck as a scientist. If you want to, try to develop a new treatment for Angelman syndrome, if you don't have the preliminary data, there's no way you're going to get an NIH grant. And so the ASF funding is really critical because, the funding comes in at an early level. It could be an idea. You don't even need preliminary data because the ASF recognizes that, you need to put money down to move an idea forward. It allows the scientist to get preliminary data that can then be used to support an NIH style grant or other grant. So the money that ASF provides to scientists is leveraged ultimately to do much more than would otherwise be done.
Yeah, ditto that. I mean, I'd add that, I think my colleagues and I have all been rejected at least once by the Angelman Syndrome Foundation for our grant applications. Like, what's beautiful about that is it makes us go back to the drawing board and try harder. I'd also add that the foundation kind of introduced me to the families affected by Angelman syndrome, and it gave myself and my lab mates the real motivation for why we do what we do. And I think seeing your beautiful kids every day through social media and at meetings and things like that just reminds us really of why we do what we do, and being able to carry out our work with a sense of purpose is huge. And the foundation gave us that too, in addition to the crucial early funding and funding for risky ideas.
Well, you guys are incredible. Thank you for spending 15 minutes of your time with us today. Two things before I forget. This is a conversation that can be had with any of you out there in the community. There is an auction item currently up for bid for a Zoom date with this crew. You can ask any questions, no holds barred, anything you want to ask. Maybe I should clear that with you guys first. But, literally, a Zoom date with these guys is on the table. So get out there and bid for that, and then go out and thank your tribe of Angelman warriors who have donated through you to the Angelman Syndrome Foundation. Thank you to our families who endlessly support the Angelman Syndrome Foundation, because these are your donor dollars at work funding research that has gotten us to where we are today in 2020. So thank you to our researchers. Thank you to our community. And I'll look forward to see if you guys pop up later in the week in other activities. So thank you.
Thank you.
Thank you for your support.
Thank you.